Welcome everyone to the ECCB workshop 2022 on integration of large-scale data for reference genome development and biodiversity. The format of the workshop is we have three speakers, each 30 minutes, and then we have a break, short break, and then we'll have a practical session on how to run the assembly. So I hope all of you hear us well and see us well. And if you have any questions, just write it in the chat box and we'll try to help you. So welcome again to this workshop session. I hope you have a good learning experience and a fun session here. So let's get started with our first speaker, Guji Zhang. It's my great pleasure to introduce Guji. He is a chair professor at Zhejiang University and center director at the Copenhagen Universities. His research interests includes developing and applying comparative genomic tools on high throughput omics data to address classical questions of diversification, adaptation, and species across a broad spectrum of organisms. He is a world leader in many large-scale international projects such as B10K, Genome 10K, as well as the Earth Bio Genome Project. Guji, it's our great honor to have you here, and please take it away.
Thank you, Shibas, for your nice introduction, and also thanks for the organization of these workshops. So thanks, Shibas, for your nice introduction again. So, well, for this workshop, because this is really focused on biodiversity, so integrate with the bioinformatics, but my background is on evolution biology, so I'm not considering myself as bioinformatics. So my topic, my presentation will mainly focusing on biological parts or evolutionary parts to introduce how we use the genomic data to transform to answer the question in biodiversity fields. So everyone knows that for many years with Human Genome Project, and after the development of Human Genome Projects, the genomic technology has been widely applied in various not model organisms. So as you can see, it's a lot of publications since 2003 that working on various different animal species. So this is just part of my my group's research interest here. So one of the central questions for biodiversity study is to use the genomic data to reconstruct the tree of life. Of course, this is kind of a dream for all evolutionary biologists to construct a digitalized tree of life using full genome data, and basically because the tree data actually is very crucial for many of the biodiversity equation, like to looking at the speciation process, adaptive mechanism, and tree evolution, and all this is relied on an accurate trace. But construct a phylogenetic tree is often very challenging, that particularly because we can see is that when you're using the full genome data, different genes that has its own evolutionary history, so it's very often that we can see incongruence between the gene trait and species traits. And there are three major evolutionary events that could contribute on this incongruence. For instance, convergent evolution, let independent species, distant species independently occurs the same mutation, and that would produce converge independently evolve with the same phenotypes. Or the other way is is based on the hybridizations that distant species with hybrid against, and some of the genes that you can see gene flows from one species to the another species that could also contribute to the gene trait incongruence with species trait. And today I would talk small about the another events that has often been ignored, that is called incomplete sortings, where simple words like incomplete sorting we can explain in ways that where the ancestor polymorphism is randomly fixed in the descendant species. So, which means that the descendant species could randomly inherits one of the genome types, the ancestor lineage. So which this would lead into that the inheritor of the same phenotype or same genome types of the distant related species. For instance, that the human and gorilla, we share about 50% of the human genome with gorilla. So, as I say, is that the contribution to hybridization that is very well known is that the hybridization between the modern humans and Neanderthals or Denisovans that has contributed that is leading to the some gene flows between this species, and also the heliconias, butterflies is also very classical examples of hybridization. But when you're talking about incomplete sorting, actually, this is basis in coalescence that is only recognized very recently because the development of coalescence theories. So it basis on coalescence theories, we can see that the polymorphism or the different ideas in the population that we can infer the common ancestors in certain times. So the coalescence terms of the different ideas really basis on the population size of the species and also the diversities of the two ideas. So in some cases, some of the ideas that can trace in back the common ancestor very long time ago, before the speciation events. So here one example is that for some of the polymorphism, we can see that some of the genetic variation that is attributed by the develops or evolve by lineage-specific ways. But for some of the idea or the polymorphism that we can trace it back in even longer time ago, that for some the coalescence times actually is or the coalescence process actually conflict with the speciation process of this of the speciation process. This is one example here is that for some for the grains and the the pins ideas that we can see is that they are coming that the coalescence time is occurs very recently. But when you're talking about when looking at the ideas in the A and B's, there's for this idea that we can see that the coalescence time is different. Coalescence history is different with the speciation process. So this phenotype is called incomplete sorting, and basis on the genomic information that actually we can infer this history for each of the individual loci in the genomes. So this is one example has been reported before, that in the gray apes, that when you're looking at the human genomes, at least 50% of the human genomes are more close to the gorilla, which is contradicts with the speciation process that we know in the gray apes. So for the incomplete sorting, then this leading to two important questions. That one is that how frequency is that the incomplete lineage sorting would occur during the macroevolutionary process. And the second important question is that where we can see this kind of a genome ties coalescence process, but if incomplete lineage sorting occurs on the gene loci, whether that could also affect the gene and the phenotype. Assumes that the gene is also contributes to some of the phenotypes developments. So, and to looking at these two questions, so one is that we need to identify some significant some lineage that has significant proportion by in community sorting occurs. So we focus on the marsupial here in these studies because the marsupial has experienced a very rapid radiation, which means that within very short times that there's a lot of marsupial lineage has been evolved within very short periods. So and previously there is also some controversies between over the marsupial phylogenies, and most of the debates focus on this single group. This group, which represents by one single lineage, is called Multiassembly, which is species that distributes in South Americans. But it's very often that this species has been considered as Australian species because their morphology looks quite similar to the marsupials in Australia. So here's some examples that where we can. So even though this species is called Multiassembly, so this species distributes in South Americas. But from morphologies and some of the physiologies that we can see that in many features that this species is much more similar with the Australian species, particularly for those species that like the wallabies or koalas groups. So, for instance, that for this species that is quite similar with the other marsupials in Australia, that it has the unpaired sperms. But the marsupials in South America that they normally have these paired sperms that in the testes. So this species also does not have the mammalians glands in the males, just like the Australian marsupials, and the anchor structure is also more similar between this species and the Australian species. So from the morphology part, there are a lot of the morphological features that has classified this species as close related species to the Diprotodontids, the wallabies groups, the Australians, one of the Australian groups from the morphologies and mitochondries. So, but instead that previous studies that using the transposon element, so the transposon element that is randomly inserted in the genomes, so it's very often considered as a very good phylogenetic signals to identify to construct the phylogenetic relationships. So what they find is that they try to identify the transposon insertion and deletion event are present and absent across the marsupial species, and they find out that where many of the transposon elements are actually supporting that the Multiassembly these groups, the South Australian marsupial species actually is the sister groups of the common ancestor of Australian marsupials. So, which means that this phylogenetic relationship is different with the mitochondria or some of the morphological trace data. So you can see that there's some controversy here. So to resolve this, that we use the whole genome data to reconstruct the phylogenetic relationship. So we construct the whole genome alignment across all the species, some representative species, using options as null groups. So based on this whole genome data alignment, so we found that we can produce a very well supported phylogenetic relationships that is putting this species as the sister groups of the entire Australian marsupial groups. So, which means that this is actually supporting the TE data that we see, but also different with morphology data. But then if we split the whole genome alignment data into small windows, sliding windows, then we can see that where there's a lot of incongruence between genomic loci. So, so basically is that we can observe three major phylogenetics relationships using different proportion of different genomic regions. So some of the genomic region actually supporting the whole genome trace that listing this species, the Multiassembly monkeys, as the outgroups of the entire Australian species. But some of the genome region actually grouping together this Multiassembly monkeys with one of the Australian groups or the other groups in Australians. So we can see that where there's a very close proportion of three topology supported by almost equal proportion of the different genomic loci, even though the whole genome tree actually receives higher proportion of supporting. But actual data, we can see that this ratio even smaller. But when you're looking at the racial transposon element insertion, actually is that it's confirmed that this is better suppose that the Multiassembly monkey is the outgroups of the entire Australian marsupial species. So, but there's another two topologies as equal proportion of this supposed by the T data. So, which means that the entire genome, when you're looking at the entire genomic region, there's a high proportion of the genome in congruence with the species trace. So, as we say, is that there's some three major hypotheses or possible evolutionary events that could that might explain this incongruence. We can exclude that this is convergent evolution because convergent evolution could not occur so pervasively in whole genome levels. But then the question is that how can we define whether this is classify whether this is contributes by the community sortings or hybridization. So basis on the evolution process of these events, we can enforce we can distinguish these two events based on the divergence times of the data. So based on the screeners of the in-community sorting, is that because we know that in community sorting should the coalescence time should occur far before the speciation process. So therefore, that the coalescence time here is the TI should be much older than the speciation time between the A and B. But instead, that if this is the contributes that by the hybridization, that's putting B and C more closer, which means that the hybridization need to occur after the speciation times. So, which means that the divergence time between these two species is much younger than the speciation times of A and B. Okay, so based on this principle, this information, actually we can classify. We can test whether the coalescence times or the divergence times is younger or older than the divergence time between A and B. So if the time actually is older than A and the divergence time between A and B is then, which means that where it's more likely that contributes in community sorting. But if this is younger, which means that it's more likely to be hybridization. So therefore, that we can, because the whole genome data can split into three data types based on the topology that they support. So we can first use the genomic regions that supporting the Multiassembly as an outgroup of the entire whole Australian group. So we can call it, this is the type, the number one trace. And this is the trait 2 and trait 3 is supporting the alternatives topologies. So based on this, we can enforce the divergence sequence divergence terms based on this different proportion of data. So the T1, which means that that there's not any hybridization occurs on this region, or the in-community sorting occurs in this region. So the divergence time here can be considered as very close to the divergence time between these two species. While the other topologies that when looking at the divergence between these two groups, or these two groups, so it either represent the collection times or either represent the divergence time after the under the hybridization. But all what we found here is that the T1 and T2, these two times, are older than the speciation time that we estimate here. So which means that it's more likely that in-community sorting could experience the current pattern that we observe. Okay, so based on this screeners, of course, we can also use other methods to test all this. I will skip that these technical details. Another evidence is supporting there's less likely to be due to the hybridization because based on geographics location and the continent's place location, that we can exclude that this is also likely due to the hybridization because these three continents have been spread already before the speciation, the divergences of these two species. So it's less likely that the species in Australia, in Americans will come back again to Australians, or the Australian species will go to the South Americans. So this could also provide additional evidence to suggest that hybridization is not possible to cause these patterns. Okay, so the second question is that whether the in-community sorting would contribute to morphologic evolutions. So assumes that there's ancestor polymorphism, genetic polymorphism that contributes to the phenotype polymorphism in the ancestor population. So in the screen up, so we love the in-community sortings that was these A and Bs would evolve with some phenotypes. But under the in-community sortings, that where the B and C would inherit it's possible that B and C would inherit the same genome types and leading to the production of the same phenotypes. So then when we're looking at the morphology data that we can collect for some of this species, now you can see is that even though this is the speciation process of the marsupial species, but in fact that we can see some of the morphology data actually supporting showing that that Multiassembly looks more similar with the wallabies or koalas, for instance, like the humerus structures, that the curve pattern is more similar between Multiassembly with these Australian groups, and also is that the vertebrae, the spines length, this T1 and T2, that we can see is that the length of this first two spines actually is more similar in this three species, but both the options and the these two other species, that we can see that T1 is much shorter than T2. And we can also some other morphological features that actually is indicating that there's some similarity between this species with the Australian groups like this. So the next question is that we want to answer is whether those genes that are affected by the in-community sorting that could attribute some of the morphology pattern we can see here. So to do this, to involve this, that first we identify those genes that has been seriously affected by in-community sortings. For instance, like we can identify those genes that have much higher similarities between the marsupials and the one of the groups. For example, this is the wallaby groups that is more similar with the Multiassembly species. They have showing much higher amino acid similarities. We can also infer the genomic loci or genomic blocks that's supporting this in-community sorting events. So here's one of the genes that we identified that is called this gene actually has a large proportion of this gene actually is under the screen of the in-community sorting and supporting that this species is most closest to the wallaby groups. So here's that we can see that where some of the amino acids that we can also find is that also supporting this screeners that like the Multiassembly has Q loci, and the wallabies and koala also have the Q loci. And the other group, the rest group has the R loci. And this is same for the other genes that we can see this patterns. So we first try to screen all the potential genes that contributes to the to vertebrate patterns, because we are interesting. Like we want to identify some of the genes that are related with this pattern. We first try to screen all the potential genes that has to be involved in this vertebrate patterns based on the gene knockout database in mouse. So this gene, or the alternative name is called GASP2. That's been found, been reported before, that knockout of the genes that change the vertebrate patterns in mouse. But to confirm that whether the in-community sorting really contributes to the morphologic pattern we observe here, is that we need to change. We designed an experiment to change the to modify the substitution here. We can see here that the substitution pattern. Here is that the wallabies and monkeys and koala both have the Q DS, and that's the T1 and T2 have the equal length. But in the Tasmanian devils and the other the groups, and also the option that has the R genome types, and that the T1 is much shorter than T2. It's very interesting that this loci actually is also very conserved, that in mouse also has the Q adheres. But in mouse, in the wild types, that the T1 is much longer than T2s. So we assume that if the this genotype really contributes to the phenotype that we observed, we could expect that if we change the Q to R's, then we expect the T1 should be reduced, or then the length of the T1 should be reduced. So, and exactly that if we perform the transgenics in mouse, that to change the Q to R's, we can exactly see that T1 has been reduced, the length of the T1 has been reduced. And this is a very significant pattern that we can observe in the transgenic mouse. So it confirms that where the genome types that it inherits in these different groups, that actually is, that could contribute to the morphologic pattern that we can observe in the current species that we examine. Of course, the people would also argue that this is only very one loci, but how about the whole entire gene? So, for instance, if the genes that the entire gene is being affected by in-community sorting, that what are the replacements of the entire genes could also affect the morphology patterns. So here that we also design an experiment that where this gene also showing supporting the closer relationship between the monkeys with wallabies. So we designed an experiment to perform the transgenic genes. So we replaced the mouse genes with the marsupial, the option copies, the orthodox, and then here we and also that we can replace the mouse genes firstly with these three genome types, the option and wallabies and devil phenotypes. So we can see is that where replacements of the genes with this three copy actually can separate the morphologic features. So we can scan the bonds and looking at the morphology pattern, and they can really separate the three species based on genome types. And here we can see is that if we replace some of the amino acids, so some of the amino acids that is bringing together monkeys with the wallaby, so we can replace the option genes that with 4 amino acids to the Multiassembly genome types. So we can see that where the chance of this genome types could could introduce the phenotypes that bring these two species together. The Multiassembly looks more similar with the wallabies genome types. So this suggests that where replaceability in high genes would also affect introduce these morphology patterns we can observe. So which means that well, in community sorting could also produce we call it the hemiplastics patterns that we in distant species produce the similar morphologic patterns. And the next question is because of how pervasive in community sorting. So we try to screen this kind of in community sorting pattern in many of the tassas. For example, this is Drosophila. We can see is that a large proportion of the many of the many of the lineage, we can see the high proportion of in-community sorting occurs. And this is also the same in the primates. Recently produced the whole genome data for many of the primate lineage. So we can also see that a lot of lineage actually has very high proportion of in-community sorting pattern. That also include the human and gorilla human and gorilla lineage, but we can also observe this kind of in the community sorting patterns in other nodes. For example, in the also in the very early branch of these groups, we can also observe a very high in community sorting levels. So here's bringing the conclusion. We can show that what community sorting actually is a very important source for the phylogenetic conflicts, and in community sorting could also contribute for the morphologic evolution. And in community sorting could also occur very frequently on the speciation process. And all these findings could only possible after we produce the whole genome data across many species. So I will stop my talk here, and I would like to thank my group people, that Feng Shaohong, who is the first author of these papers, and Michael Schraps in Aarhus University have to develop the tool to detect in community sorting signals in the whole genome data. And I would also thank my collaborator in Australia who provides support on getting the samples and some of the morphology data. Merlin, Renfrey in University of Melbourne, and Christoph Hagens in the Australian Museums. Right, thanks again for your attention, and I will be very happy to answer the question. Thanks.
Thanks, Guji, for the nice talk. So, any questions? People can raise hand. Maybe I can start. Guji, what type of whole genome sequencing is done for this analysis? And if you can briefly tell some of the steps of the bioinformatics pipeline.
Okay, well, because this is a very very ancient project, a very old project that's organized in my groups. We use the short range basics genome assembly for many of these species to do this analysis, because this project has been initiated probably 7 years ago. At that time, there's no long range sequencing reps yet. So, but yes, it's very important that if we can use the long range sequencing, we probably could also increase the detection powers of in-community sortings, because we can see is that we only identify 569 megabase of whole genome data, and this is only probably 20% or less than 30% of the genome size of the mammals.
And are there any recent studies on these species based on long-read sequencing, and if there are any controversies that have been resolved already, or there are any still the same open questions remain?
Well, I don't really think so. So, well, if we use the long-range sequencing, we could probably we can identify more regions that to construct the phylogenies and also identify the intermediate sorting signals. And we could probably also identify small transposon elements that also could aid for the phylogenetic analysis. But I doubt that the overall pattern would change with the long range technologies.
Okay, thank you. So if we don't have further questions, let's thank Guji for joining us today. It was a lovely talk. And then we move on to the next one. So due to COVID, Jean Myers is not able to join us today, but our next speaker uh, is Danang. Danang has recently finished his PhD from ETH Zurich in animal genetics, and he's interested in automating whole genomic data analysis with reproducible bioinformatic pipelines. So he has extensive experience and interest in more representative reference genomes with the inclusion of the genetic diversity. For this purpose, he integrated millions of population-specific bovine genetic variants with current linear reference to create a comprehensive pangenome graphs specifically for bovines. So I'm delighted to have Danang here today with us. And Danang, please take it away.
Thank you so much for nice introductions. And also thank you for the invitations. Today I'm going to talk about my PhD project on establishing bovine pangenome graphs. My name is Danang and I did my PhD at ETH Zurich. So let's start. This presentation will consist as follows: start from introductions and the objective of the research. Then I will continue with result and discussions. And then finally, I will come up with conclusions and outlook for future research. So let's start for the introductions. Reference genome is useful for many genomic analysis as we all every genomic analysis will depend on the genomics on the genomic reference sequences, such as for alignments, for comparing variation. We always compare relative to what the reference sequence. However, reference sequence currently was built on single or few individuals. This applies in many population, including humans, also in cattle. For example, in cattle population, the reference genome was built from single animals called Hereford, shown in the left figures. However, the genomic sequence might not be representative for whole cattle population. As you can imagine, that cattle exhibit high genetic diversity because they breed for different purposes, and they diverge because of this divergence into hundreds of distinct subpopulation or known as breed, as shown as in the right figures. As I mentioned before, that linear genomes, when you represent collapse all sequence diversity into a single nucleotide in its positions, it's unaware of genetic variations. That it cannot sufficiently represent complete species diversity. This might be problematic if your species that you compare, or your species or individual that you are compared quite far from the reference sequence. And this all called reference bias because all of the genomic analysis will be biased toward the sequence which are present in the reference sequence itself, even though the sequence is not the most representative individuals or the most ancestral or the most used individuals. And
Excuse
this
me, but
reference,
are you changing your slides? We don't
mm,
see your slide
yeah,
change. Or
yeah,
if
I changed it.
or it's just me, but
Yeah, could you see soft reference bias here?
yeah.
Yeah,
So we see the slide linear genomes are unaware of genetic variations now. So is it
yeah.
in a slide presentation mode?
Yeah, I am doing it in presentation modes. And could you see these linear genomes do not sufficiently represent complete species diversity here?
Yes. Yeah.
Yeah, and then prone to reference bias. Yeah,
Yes.
next.
Yeah.
Yeah,
It's
yeah,
good. It's good
yeah.
now.
Thank you. No worries. Yeah, I will continue. So this limitations, inherent limitation of reference genomes of the use of single reference genome called reference bias, and this divided into two major subproblems. The first one is so-called soft reference bias. For example, if you compare sequencing rates which are quite diverse from the reference, this might be more support for rates which are supporting the reference allele, and less support for rates which are diverse from the reference. So if you have like heterozygous genotypes, there will be support, where more support for the reference allele. And if there are multiple variations, that these routes might be misaligned because of suboptimal alignment scores, or it's even indicators. And the second problem is so-called hard reference bias. Sorry. That reference genome does not contain large structural changes that diverge in the populations. For example, these reference genomes does not contain this large insertion here, so that we cannot assess genetic diversity inside of these missing insertions, and this so-called hard reference bias. And there are multiple efforts for creating more representative reference genomes, and this is first because of currently genomic resources are generated at rapid rate. This including long sequencing rates, because they are more affordable, and there are needs to make more the reference become more representative, and this drives the concept of pangenome. So move from genomics into pangenomics. So the idea here is that we use collection of genome as a reference rather than collapsing all of genetic diversity into single reference. So the idea of pangenome is that in this collection of genomes, you have a part which is shared in all of member of population, so-called of core genomes, and there's also part which is shared, which is dispensable. They're only present in some part of samples, or private in some of the samples. So there are multiple efforts to represent this genomic data, and one of the promising solution is by representing all of this diversity into so-called graphs. So the idea here is that we can use graph to represent this allelic diversity in compact and coherent reference structures. Yeah. So I will. Sorry for this.
Maybe let's start from like before graphs. Yeah,
Yeah,
this is good.
yeah,
Yeah, now
yeah,
all
yeah,
set.
yeah, yeah, yeah. So I mentioned first one in the first slide, that genomic linear genomes, where you collapse all variation into single representations, cannot sufficiently represent complete species diversity, and this cause problems, so-called reference bias, that all of genetic comparison will always be biased toward sequence choice of the reference genomes, even though the reference animal or reference sample does not represent the full representative individuals, or the most representative individual into populations. And this divided into two major subproblems. The first one is so-called soft reference bias, where if you have sequencing rates and you compare with reference, and you have variations that might be less support if your rates containing variations, and more support if the rates match the reference. And if there are multiple variations, there might be suboptimal alignment scores that result in misalignment, or rates that cannot be aligned altogether. And the second one is called hard reference bias, that the reference genome does not contain like large structural changes, for example, these large insertions, and thus we cannot assess genetic diversity inside of these missing segments. And this drive toward creation of more representative reference genomes. The other drive is because genomic resources are currently generated at rapid rate. This includes short sequencing rate and long sequencing data. They are now become more affordable, and there is need toward creating reference genome which is more representative. And this also a drive to the concept of pangenomes. And the idea of pangenome here is that we rather than collapsing all genetic diversity into single structures, we use collection of genomes to represent genetic diversity. So the idea here is that based on this collection of genomes, we have so-called core genomes, where this segment of genomes present in all member of populations, and dispensable genome in which that some part of this genome present only in few or privately in some individuals. And there are multiple ways to represent this data, and one of the promising solution is by using graphs that can represent this allelic diversity in compact and coherent reference structures. For example, if we have these four samples, we can collapse regions without any difference into single nodes shown here in the blue arrows, and then we can add this variation as alternative nodes in the graphs. And because we know the input sequences, when we walk toward the graphs, or we call it traversing the graphs, we can derive the original input sequences from which the graph will fit in. So for example, we can add path of the current reference and path for the other three samples. And there are two major genome graph implementations. The first one is so-called variation graphs. So the idea here is that we use current linear genome as backbone of the graphs, and then we add variations which previously catalogs, and this creates so-called variation graphs. And we can take the variation from previously catalog variations, for example, from feature files. And there's also there's also so-called multi-assembly graph. Even though variation graph is simple, there might be miss variation which is previously cataloged from maybe only from sound sequencing rates, which is miss a lot of sequence variations. And the idea of multi-assembly graph is that we constructed graph from multiple genome assemblies. So the idea is that we perform multiple genome alignment of this assembly, and then we induce a graph from the multiple genome alignments. So this is more powerful than potentially containing all of genetic diversity, including large structural changes, as I mentioned before. And then objective of my PhD is that I assess the utility of graph genomes for sequence variation analysis in cattle populations. And this talk, my PhD containing three parts. The first one is variant genotyping from local variation graphs. And the second part of my PhD is construction of population-specific and multi-population variation graphs. And the part three is that we try really to create like multi-assembly pangenome graphs. So the idea is we different graph implementations and assess its utility for sequence variations analysis. So for the first part is what I did is that we did variant genotyping from region-specific graphs. So this is based on the software so-called graph typers, and this we create like graph-based variant discovery pipelines. Then we use it to genotype like samples that we have in-house. So the pipeline itself consists of two different iterations. The first one is so-called discovery. So this part is similar like when we did variant discovery from linear genome. So the idea is we align reads toward the reference, and then we call variation from the aligned reads toward the reference. However, we perform second round of genotyping, so-called refinements, where we break down these genomic regions into windows, and then we collect variation in these windows, and then we construct specific region-specific local graphs from these variations, and then we took previously aligned reads and then align these reads again toward these specific local graphs. The idea is that we can refine the alignments toward these local graphs. And then we, at the end, we perform genotyping from the refined alignments. And then using because we don't have like genome in the bottle as Truth said, we compare this with SNP arrays like orthogonal methods to assess the accuracy of the genotype callings, and then we compare it with current standard of genotyping using linear genomes, and this graph paper pipeline has higher performance in multiple metrics, even though it does not increase that's a lot because SNPs and small variation, basically they're already quite high using sorted data, but we see a lot of improvement in when we look at the Mendelian consistency on indels. And this result is quite promising at the time because we don't use full graph analysis rather than we refine alignment on region specific graphs. And this pipeline is competitive with linear based tools. For example, we can use this genotype likelihood output from the pipelines to do genotype imputations, and to do genotype imputation of it, and then we see similar performance in even higher performance than current standard tools. And then we also see that this pipeline does not require more computing requirement than current standard tools. So this is quite promising. And then we move into second part of my PhD, which is construction of population-specific and multi-population graphs. In the first part, we only augment or add variations into the graph from sample, which is part of the graph. So we don't use external variations. So in the second project, we try to also consider external variations because we already a lot of previously cataloged genetic variations. So for this purpose, we also use variation graphs, but we do this using previously cataloged small variations that has been cataloged in 4 European cattle breeds, shown here in the figures. Based on this PC analysis, there are 4 genetically distinct populations. And then again, we do almost similar stuff like before, where we investigated rate mapping, variant genotyping, and allelic balance from the graphs. And first analysis that we do is that if we have external variations, the first question is what kind of variation that we need to put into the graphs. So whether it's advisable to include all variation or we need to select some variations. And then the result shown here is a variant selection when we look at the allelic frequencies, where we simulated reads toward the graphs and then filtered variations that we include into the graph based on allele frequencies. So if we look at the graph below, and we go into the left directions, meaning that we include more variation with lower allele frequency. As we see here, that the mapping accuracy increases, so as lower mapping error as we add more variation into the graph, indicating that if we include more variation in the graph become more informative. However, this does not sustain the increase of accuracy. Actually, the mapping accuracy start decrease or even does not improve at all or even decrease when rare variations added into the graphs, because they are rare, only present in some population, and inclusion of this variation will just increase the complexity of the graph without making the graph become more informative. So there is also differences in mapping accuracy across population. If we construct graph from 4 specific populations, their performance of the graph is different. Is different compared with other populations. For example, this one is like original brown V here, the green line, and this has a higher mapping error. And when we check, this also has the higher genetic diversity. So the second question that we ask is whether it's advisable to include population specific. So we create like multiple population specific graphs, or we can combine all of the population specific graph into so-called like unified pangenome graphs. So for example, for this purpose, we also do a lot of simulations toward mapping of this population specific or multi-breeds, or we include like variation without any considerations, that we took the variation from SNP database without any considerations and randomly put variation into the graph from the SNP database. And we see that if we compare the brown box with the red box, graph mapping with pre-selected variations is always more accurate than linear mapping, indicating that variant selection is important to be considered for graph constructions. And then, as expected, that the most accurate mapping achieved with population-specific graphs because they are specific for your populations. However, the mapping towards the pangenome graph is almost as accurate, indicating that the construction of unified pangenome is possible that will be applicable toward a lot of breeds. So we can use single graphs toward more populations, and its performance almost the same. Then we constructed multiple population specific graphs. And then because all of the previous analysis were based on the simulations, and then we look at when we use the real data. So we used previously variant selections to select 14 million preselected variations, and then we created like this whole genome graph for cattle. And then we assess this mapping and genotyping performance on sample which is not part of the graphs. And then when we look at the alignments, we see that there are a lot of like more than 10% increase in a perfect alignment, meaning that this sequencing rate matches perfectly toward the path in the graph without any differences, meaning that the graphs now represented fully in the sequencing rates. This when we compare with the linear genomes. However, we see slightly lower in part of the unique alignments. There's also downside when you have graphs, you have a lot of multiple paths that you need to consider. That this might be increased like ambiguity that reduce the proportion of unique alignments. And then we look at the genotyping performance. In this result, I showed as the genotyping allelic ratio at the heterozygous site. Imagine like if you have heterozygous site, you have support for reference and alternate alleles. And if you have reference bias, the more support for the reference allele than alternate. So the allelic ratio will be low. Shown here in the figures, as you goes down here, the bias become higher toward the reference. And when we look at the bias across variant lengths on here, when we look at the linear analysis, we see that as the variation gets longer, the more bias is observed in the genotypes and in the heterozygous genotypes. For example, here, which is much worse if we see large insertions, because the sequence simply not present in the reference. However, when we look at these whole genome graphs, regardless the variant length, the allelic support is close to half, meaning that both reference and alternate, they are equally supported, and there's no or the bias can be reduced dramatically. So this is the second part of my PhD, and the third part of my PhD is instead of only using small variations, we try to establish profile multi-assembly graphs. So the idea here is that we have collection of multiple genome assemblies, and then we try to unify this assembly into pangenome graphs. We use implementation of graph from mini graphs. So the idea here is that we created these relationship trees based on these assemblies, and then we use current reference genome as a backbone of the graphs, and then we build the graph iteratively using information from these phylogenetic trees. So we add the other tree genome which is closer toward the reference, and then we iteratively align this assembly to the graph and add new variation into the graph, and then at the end we also add the most divergence from this reference. And then for this purpose, we also generated one assembly for this original brown V, and this original brown V data was constructed using latest long read sequencing technology of HiFi, which resulting in higher contiguity and completeness compared with other assembly which is based on the old technology of long read sequencing. and then after we build the graphs, and then we try to look at sequence which is not present in the current reference sequence. We call so-called non-reference sequences. However, implementation of mini graph does not keep path information. So we need to find a way to assign nodes with original sample in which that these nodes constructed. So for this purpose, we develop like new methods that where if we have graphs and then we have assemblies, we can try to assign its nodes to what the origin of the assembly is. So we align back this assembly, and then we traversing this alignment, and then we label its nodes with the information from the alignment. And then from this we can enumerate path in the bubbles. So I haven't talked about the bubble. So bubble is regions in the graph in which that it's containing variations, and it diverts path in the graph where it containing variations.
Okay, maybe let's wait 2-3 minutes. He will be able to rejoin. Yes.
Yeah,
Okay.
yeah. Can you? Sorry. Yeah, sorry, sorry. I think I can share my window. Yeah. Can you see my screen now? Yeah, yeah, yeah. From where should I repeat this presentation? Yeah, I can
Good.
I can start from this, right?
Yes.
Yeah, yeah. And based on the methods that we use to assign sample labels into the graph, we can extract these structural variations from the graphs. And then application of these methods, we discover non-difference allele of SVs around 70 MBs, and this is represent sequence present in the graph but not in the current reference sequences. And then and then based on the labels that we put in the graphs, we can explore the sharing of these non-reference sequences across samples. For example, we discover around 30 MBs of these non-reference sequences specific toward one of the assembly, which is most diverse assemblies. We also found around 4 MB of sequences which present in all of samples used to construct graphs, but not in the reference sequence. And this represent might be represents deletions, which is specific toward the reference sequence. and then uh, and then uh, and then not only on the statistical part, like how many reference sequences, we also look at the functionally active elements. We look at also biological part of this non-reference sequence, where we try to look at the functionally active elements. So for example, we look at this 70 MB, and most of these non-reference sequences are repetitive elements, like 70% of it. And then we also do like computational predictions whether we can detect genes inside of this non-reference sequence. And then we perform computational prediction, and then we validate it with transcriptome data. And we found hundreds of these putative genes present in non-reference sequence, where most of these genes are shared in all of the animal, which is not part of the reference animals. And then when we look at the functions of these putative genes, many are related to immune response, signaling, lipid and protein metabolisms. This when we do like enrichment analysis. This makes sense because immune response is one of the most highly diverse highly diverse regions in the genomes. And then after we found these functionally active elements, we try to look at whether these functionally active element contain differentially expressed transcripts. So for example, on this analysis, we revisit analysis the infection biology of Mycobacterium bovis. So this is consists of transcriptome study of match, control, and infected cattle. So the idea is that we reperform this differential expression analysis on this information from pangenomes rather than using linear genomes. And then when we look at the expression of non-reference genes, like these 100 non-reference genes that we previously catalogs, we can separate infected from control cattle using only information of these novel genes. So the idea here is that we can use this information from these novel genes to separate they are informative to separate this in control infected cattle. And then we found 36 non-reference gene are differentially expressed with p less than with false discovery rate less than 5%. And then shown here in the figures on the right side. And then the top of this differentially expressed gene is related to leukocyte receptors. And when we look at these genes are not present in the current reference because it's located in block of leukocyte receptor, which is assembled in the current reference genome. But we can found these genes on more complex assemblies in the graphs, and then we can use this information for differential expression analysis, which is missed with the current reference because they are simply misassembled. And then lastly, we shows that these non-reference sequence are biologically relevant. For example, we found like 50,000 of non-reference sites that containing small variations that segregated between and within populations. This indicates that these variations this this variation inside of this non-reference sequence are informative and biologically relevant. And then when we look at the annotation of the variations, we found like thousands of variation affected coding sequence and many annotated with high impacts, which might indicate that this variation might affect phenotypes, even though it needs further investigations. And then when we use this non-reference variation from clusterings, this can spread animal by subpopulations shown here in the figures, indicating that this variation from this non-reference sequence might be informative and they are contribute for breed differentiations, which is currently missed. And finally, I will come up with conclusion of my study is that my PhD is on the application of graph-based approaches in the cattle genomes, and as I'm aware, this is for application of graph-based approaches outside of human genomes, first in cattle genomes, and we show that it's quite promising compared with current standard using linear genomes. And as I mentioned, this is a pairing strategies to mitigate reference bias. In the first part, I showed that it can lead to unbiased and accurate sequence variant discovery. In the last part, we also showed in the functionals and biological analysis that this can make so far neglected genetic variations amenable to genomic analysis. And my PhD also provide computational framework that we can use as alternative to exploit more diverse reference structures. And for the future directions, this is only the first step toward pangenome analysis. So it's more advisable to build more comprehensive graph with more variant classes and more breed and more species. This has been done in human pangenomes where they created graphs from hundreds of populations across human ethnicities, which is Silva is one of the members of this. And then I always imagine that instead of only using variation data inside the graph, we can also integrate functional genomic data like transcriptome data, epigenomic data directly into the graphs so that we can perform integrative system biology analysis from the graphs. That's what That's what I mean before. So the idea is that we can integrate all of genomic informations, all of omics data into the graph, not limited toward sequence, only sequence formations. With that, I will thank for your attention, and I apologize for all of the technical difficulties. And I thanks for all group members of Genomics Group, particularly to my professor and also Alex, which I work a lot, and then I also thanks for all parties which all of institute which supported my research, and also for funding for my PhD, and also thanks for many people which made this data and software publicly available. Thank you, and I'm happy to take any questions.
Thanks, Danang. Any questions? Maybe I can start.
Yeah.
So, using this reference-free pangenomics, how much more structure variations have you found compared to reference-based pangenomics? Like you mentioned about minigraph and VG,
Yeah.
but and without reference, like you purely work through the assemblies, then how much more? Do you have any approximate? I,
Yeah, so this is also topic of my current small projects, in which that we compared reference based pangenomes and reference free pangenomes, right from cactus or PGGB, and we found a lot of, because with reference free you are limited toward the quality of your backbones. For example, this RSUCD, which is cattle reference genome, does not contain like complex variation, so you miss a lot of variation in these complex variations. And with approach like cactus or PGGP, you are basically do all versus all alignments, where you can catalog all of genetic diversity irrespective of the quality of the backbones. And using cattle, if you see in cactus and PGGB, they can provide like, currently it's like my approximation is around 10% more SPs, which is present in complex regions, which is not present, or the assembly quality is not good in the reference backbone. But it depends on the species that you are talking, that you are analyzing. That's my opinion.
yeah, thank you. Let's thank Danang
Yeah,
for
thank you.
nice talk. Okay, so I can go next. I'm Shilpa Garg. I'm an assistant professor at Department of Biology in Copenhagen University. So today I'll give you a tour on new methods for chromosome scale assembly, specifically using the long accurate reads and high C uh, technologies. So first, what's chromosome scale haplotype resolved genome assembly is? So as we know, like humans or other uh, animals or plants, they have more than one or more than two copies of each chromosome. For example, here you see these two copies. And advances in sequencing technologies, specifically the PacBio Hi-Fi and the Oxford Nanopore Q20 plus chemistry, has given us read lengths in the order of few tens of kilobases, and they're highly accurate reads, like about error rates are only about 1 to 3%. And next, but these reads are still not long enough to span the whole repetitive region in a genome. So that's why we further require newer long range technology that is known as high C. And I'll go a bit more in detail what these technologies are and what type of data sets we get from these technologies. So the next question is, how can we combine these new sets of data types in a unique bioinformatics framework to produce the haplotype resolved genomes on the chromosome scale level? And it has many applications, as like Gucci and Dunning have presented, that it helps to identify genetic variations that point to high susceptibility to environmental stresses, and also helps in evolutionary and comparative genomics. So, a bit more into the hi-fi technology. For example, it starts with a high molecular weight DNA, and then you ligate the adapters. And this leads the primers to be annealed, and the DNA is polymerized. And then you get this circularized DNA, which is now ready for sequencing. So essentially, you then do these repeated passes of this circularized DNA, and then call a consensus on these sequences. And this produces high accurate reads as well as highly like long in the order of few tens of kilobases, tens of thousand, tens of kilobases. Yeah. And then in the high C, it's essentially, it captures the closely spaced segments in a space. For example, in a 3D space, maybe this thing is quite near, but when you stretch it on a linear space, it is a few tens of megabases apart from each other. So actually, HiC is able to capture those HiC contact points, and that we know that they are coming from the same chromosome and the same copy of the chromosome. So without going in detail of like how this technology work, which is out of the scope of this workshop, the main take-home message from this technology is that, is if you see a plot on the right axis, on the x-axis is genomic loci, and on the y-axis is the high-seq contact maps. So darker the color is, higher is the frequency of the contact points over the genomic regions. So for example, here if you see closely in this plot, um, so you see a darker red color in about tens of megabases of region. So this suggests that you can get high long range interactions up to 10 megabases, but then if you go further apart, you see the signal becomes more lighter and it's more erroneous data. So the next question is, how can we combine in a bioinformatics workflow to produce the chromosome scale haplotype resolved assemblies? So initially I started with humans and they have low heterozygosity rates. So we proposed this new workflow, which is to start with hi-fi data and produce unphased contigs from them, and then scaffold those contigs using high C to get these sequences. These are on the chromosome level. And further, we remap the hi-fi reads to these scaffolds to call these heterozygous SNPs. And these heterozygous SNPs are informative now to separate the reads to two sets that represents the haplotypes. And finally, we assemble these haplotype sets to get these phased contigs. So when we did this workflow, because of the advancements in these hi-fi data and also the algorithms behind that, we for the first time can now produce these humans like genomes, like about 3 gigabases, and diploid genomes with the low heterozygosity in about half a day. Then we were also interested to scale up these methods to other genomes with different heterozygosity rates and with their different repeat content. So in this case, where actually this workflow may break because it has this collapsing step of haplotypes, which may seems to be trickier for the highly heterozygous genomes. So to overcome that issue, here we propose a new workflow, which is actually a graph-based approach. The main idea is to use phasing information in the overlapping step. So we find the overlaps between of the long accurate reads, and this workflow, this algorithm is actually designed specifically for long accurate reads. And within the overlaps, we find these SNPs. And if the characters the alleles on these SNPs, they are same. That means they belong to the same haplotype, otherwise they belong to the different haplotype. So we encode phasing plus overlapping information in the form of a graph where each node is a read and the connections are now the haplotype specific overlaps. So we demonstrated this method initially on segmental duplications. Then HiFiasm actually implemented this workflow for the whole genome level, and we use HiFiasm to get the Hi-Fi unitic graph. And then on the graph space, we bring in the Hi-C data. On the graph node sequences, we map the Hi-C reads to the graph. The, the nicer, the the good thing about mapping on the graph is that now graph contains all the heterozygous information and any type of repeat, so it enables better mapping of Hi-C reads, as well as this Hi-C mapping is now based on the k-mer based approach. So we break the sequences in the graph and also the Hi-C reads and then hash them. So this approach using k-mers turns out to be working pretty well, and also it's quite fast that you can perform this haplotype aware Hi-C mapping within a few hours, which previously used to take like 8 or 9 hours. And then based on the maximum support of Hi-C reads on these, we've tried to find the haplotype paths for each component first, and then we scaffold these phased contigs using neighborhood property that inherently captures the Hi-C correct data characteristics, and to get the phased scaffolds that are actually now on the chromosome scale level. So as we know, there are recent sequencing initiatives that at the global level, like ARGA is one of them that probably some of you have seen in the previous workshop, which aims to produce high quality reference genomes for all type of species in the Europe. And we are an active member and committee coordinator to contribute towards the reference genome assemblies through our methods. And also there are multiple other initiatives at different continents like Africa and so on. So we have already started to contribute towards these initiatives through the recent methods that we have developed. So initially we have applied on insects genomes, which have, some of them have a high heterozygosity rates, and which are, the genomes are relatively smaller than the human genome. So we observed that when we calculated the total size and continuity and QVs, we could produce comparable results. And also the time and the memory, they are relatively reasonable to be able to apply these methods on the production level. And then we also looked into some of the clinically relevant regions, for example, in humans, like HLA. So HLA is a unique and highly polymorphic region with a lot of variation. So we applied these methods to assemble a few HLA regions for a few human genomes, and then we aligned them to the reference genome. And here in each panel is a haplotype, and the darker color represents the divergence rate from the reference genome. So higher is the divergence, higher is the so darker the color is, higher is the variation in it than the reference genome. So here we observe that there are many regions like HLA-AA, and then there is one region here, even there is a variation between the haplotypes within the same sample. And then there are other genes such as DRB1 and DQ1, which has about 10% of variation than the reference genome. And these things we will miss if we do only using short reads and also do only using reference-based analysis. So, we observed a similar pattern in another clinically relevant region, which is Kier, and also it has associations to many diseases like narcolepsy and psychiatric diseases. So here we observe the similar trend that there are few regions. For example, here you see 10% divergence to the reference genome, and there are DL3, DL4, and DL2. They have a huge amount of variation between the haplotypes in each sample. So next, we were also interested to study the structure variations. So we took these assemblies and mapped them to the reference genome and called the structure variations from these assemblies and compared them to the genome in a bottle ground truth. And here we observed that we received a precision and recall of more than 90%. So this suggests um, uh, to be more precise in this plot, on the x-axis you observe the size of the structure variations. For example, on the right are the insertion, on the left are the deletions, and here you see the precision and recall. So here in the plot you see that we can now find large structure variations from these assemblies, and the precision and recall is also more than 90% compared to the genome in a bottle samples. So these were the new methods developed and demonstrated on different types of genomes. The next is, once we have these assemblies, how new regions discovered through these long accurate reads and algorithmic methods, how can we annotate them? So this workflow is basically based on the ensemble workflow. So you can take long accurate RNA reads and then map them using Minimap 2. And then you can do some sort of error correction and filtering, and then use the ORF classification, and finally collapse and merge step. So this annotation is not yet implemented in cloud, but we are still in the process of doing it. So next is, once we have many genomes assembled, how can we compare and use it for more evolutionary studies? So this is where we have a new concept known as chromosome scale haplotype resolved assemblies, which is, we imagine you have genomes from different populations, like from Africa, Australia, Eurasia, Europe, and so on. So you take all these genomes and put them into a graph pangenome, which means you can represent the different type of variation. For example, there could be this tandem repeats. So different populations have different number of copies in a repeat. So you can represent this in the form of a bubble like this, that like Danang also introduced. And then also the graphs enables the SV discovery and their associated functional workflow over the long range information. Next, also pangenomes helps to find new types of diversity in the plants, and also new cultivars that can respond to different stresses. For example, for the rice, there are different species, like at the cultivated level, or there are wild rice. So you can make uh, these hybrid breeding between different haplotypes, and you can make new cultivars that can respond to a better stress, environmental stresses. So traditionally, as Danang presented, uh, the uh, the genomes used to be, first they were collapsed, and also they were fragmented. They were not, and they were essentially a set of linear genomes, so which has the limitation of not capturing the um, full variation, and also in a haplotype resolved manner. So this is where the chromosome scale haplotype resolved methods come. So there are different types of graph pangenome methods. Initially, the field started with a linear reference genome, and then you incorporate the variations, for example, these SNPs here, to the graph, and then you get a bubble like this. So this method was quite efficient because you work on linear reference coordinate system. You go from left to right, and then you just add one by one to this linear reference. This was quite fast and quite impactful, but it has some limitation as it is first on the reference base and it doesn't capture the full variations. So the next approach is how can is you are given the chromosome scale haplotype resolved genomes, and then you construct a graph pangenome without any bias to the reference or without any bias to the variation. And to do that, there have been recent few advances like K-Keys, Minigraph plus Cactus, and there is a recent method from Spain which is quite impressive, which is base level wavefront alignment algorithm, and this can give you a quite good graph pangenome that captures the full variation. But since these comparing few tens of gigabases of genomes and comparing millions of sequences, if we want to scale up, then it's quite it's still a quite computational expensive and cumbersome method. So to address that limitation, here we proposed an hierarchical approach. Imagine you have these taxonomic classes. For example, you have this hierarchy given to you: class, genus, and species. So instead of representing all the variation in the one pangenome, we propose an hierarchical pangenome where you enable the capability to access a pangenome at multiple resolutions. For example, we start with the more with the class levels, and we take the sequences that are representative of that class. So we add a variation here, and we also index some of these similar sequences to avoid any repetition. So first we do it as a class subgraph level, and then we do at the genus level, and then we do at the species level, and then the interactions between these resolution is maintained by these interlevel connections, which is actually done through these similar sequences. So the advantage is this enable biologists to interact on the pangenome level in a more efficient manner and at multiple scales. But there is one limitation here that it, for example, if there is a species that is highly divergent, then like maybe like 80-90% divergence, in that case we will not be yet able to capture that much of divergence level into this model. So to conclude my talk for the methods part here, so I presented the latest sequencing methods like HiFi and Oxford Nanopore and Hi-C that gives us opportunities to produce a high quality reference genomes for any type of biodiversity species. And I have presented you two methods for assembling the genomes. So the first is that works very well for the low heterozygosity genomes, but the next one that works well for highly heterozygous genomes. And also I gave a brief overview of the annotation pipeline. And then I also presented like how these can be helpful in biodiversity for underpinning complex biology. And thanks. Then maybe we can take like 15 minutes break, and then we can resume our practical session, like how can you assemble genomes in cloud. Yeah. So welcome back, everyone. I hope you still hear us and see us well. So I would like to introduce June Aruga. He's one of our lab collaborators who is experienced and interested in open source development, pipeline development. So he offered to help with some of the pipelines in Widal and Terra. So now we'll do a practical session of how can we actually assemble genomes using hi-fi, like long accurate and Hi-C reads in a cloud platform. So I'll share my screen now. Okay, awesome. So the first step in the pipeline is genome characterization. For example, given you have some sequencing data, so you want to get some of the estimates. What could be the genome size? What could be the heterozygosity rate? What could be the how the plotting? So to do that, we have actually put together Gene Myers tools, FastK, GeneScope.fk, and Polyplot into a pipeline. And these tools, they are specifically designed for long accurate reads, and they are very fast. For large genomes also, you can do with those with a few tens of threads, you can do in like several tens of minutes. And then to assemble genome, we follow a very simple pipeline to first construct a graph, which is a phased unitic graph, either from HiFiasm or Shasta, depending on whether it's a HiFi data or ONT reads, and then use PS tools to combine Hi-C with this graph, and which will produce the final phased scaffolds. So now June will demonstrate a pipeline that is put together uh, to do, do this task. Um, yes, June,
Okay.
take it away.
Yeah. Thanks for everyone coming for today. My name is Jun Alga. I'm usually working for the company called Red Hat and working for open source. And yeah, this time I have the opportunity to work for this pipelines workflow. I'm happy to share the tips for you. And first, I would explain about the process of our workflow. The process is first we will use the workflow definition language. It's a language to describe a workflow. I think there are several some pipelines. But this time we choose WDL workflow definition languages developed by a Broad Institute. And there is a workflow engine. The name is Cromwell. It is the Java library, so we also need Java. And Cromwell executes WDL files. Files describe the definition of the workflow. And for the workflow WDL file, in this time we used one is main workflow WDL file, and this WDL file called sub workflow files. We have three workflow WDL files. One is for estimation. It is the first process of evaluation of the data. And the second workflow is for assembly. And the third is scaffold. And workflow can call the name a task. Task is a kind of function in the programming language. Task can describe a command line, Linux-like command lines, and also can add the Docker container. It is the running environment command lines to run. So, and in this case, for example, assembly sub workflow can refer the output of the workflow estimation. And also the sub workflow scaffold can refer the output of both sub workflows, assembly and estimation. Okay, so let's move on to the next slide, please. Silva?
Yes, I'm on next
Okay.
slide. Let me see.
Okay, I can see it. And we let me explain about the flow, some architecture of how we run the workflow. First, I like to introduce our GitHub repository. I shared this URL before this resting time, but I would like to share now for your convenience. And first, we need a GitHub repository to manage WDL files and some necessary files. And GitHub repository we need, and also we need Docstore website. Docstore is the website managed by Broad Institute. It's the website to manage WDL files. It's a hosting site. And we would like to share the URL now to the chat box here. And later you can see details on the GitHub repository's wiki page. We wrote it. And Docstore website links with Terra workflow. The Terra is the website. It's a cloud website originally founded by Broad Institute and the company Verily. It's a subsidiary of the Google. So this website is a cloud service using Google Cloud. And it is specific for biology pipelines. And yeah, what we did is first we managed the WDL file on the GitHub repository. Then we created the page, workflow page on the Docstore website. And Docstore website links to GitHub repository, and Terra workflow links to Docstore website. And as a development, we developed with GitHub repository. And GitHub repository has some main files to run WDL files on local environment. And also GitHub repository has GitHub Actions, their CI, continuous integration service to test. So as a usual case, we tested with GitHub and GitHub Actions, and then Docstore synchronizes the source code from GitHub. Then if we want to test some run the WDL files on cloud, we used Terra workflow. And I just want to mention that Terra is a very nice platform because it's cheap, right? It's not free, but it's cheap. So in our case, running one workflow is less than 0.1 USD. So it's cheap. So Silva, you can interrupt if you have something.
Yes, yeah, I agree with Jun. Like, if you want to assemble genomes like in a routine fashion, like you want to just give input data and then submit a job, and this workflow and also Terra enables you production of assemblies in quite fast and cheap manner. Yeah. Any
Okay.
maybe we can ask questions before we can really show the website. So any
Yep.
questions so far? Any questions on Terra or Vidal or any step in the pipeline?
Any question is fine. what is your hobby, for example?
So we understand that you understand the pipe, like the steps, but so far, and but feel free to ask any questions and interrupt at any moment. So this is more of an interactive session. So if you have any practical questions, just ask in between. We don't have to stay until the end too. So yeah, so let's move on then. Uh, so should we share a GitHub, like in case people wanna see like an initial GitHub, and then we can show. So do you see my GitHub screen?
No, yes, I can.
Okay.
So do we explain about the GitHub repository first, right?
Yeah, maybe that would be good. Yeah.
Sure, okay. And first we see some files on the GitHub repository, and for README, it's some bit, sorry, can scroll down. README very small. If you want to see the detail, you can click the link and to go to the wiki page. So here is just information. So can you move on to the main page again?
Sure.
Yep. Okay. So we can see some files. The file ending with WDL is the WDL file. And can you click the name biodiversity genomics WDL file? Okay.
I'm sorry. I'm
And
doing it now. Yeah.
yep, yep. And this is the actual workflow file. And the first line, we see version 1.0. That means right now, Terra only supports WDL language specification version 1.0. There is version 1.1, but we recommend to use 1.0. And we can see the files importing three WDL files. It's a line of number 2 to 4. So we are using other WDL files. I said sub workflow. So we are using other WDL files to describe sub workflow logic. And yeah, roughly, you can see some workflow, some starting with workflow, line 6. And then we define input, line 7 to 14 is the definition of input items. And we see some logics task in the other files. And line 36, we see output. This is the definition of the output items. Yeah. And can you move to the top page again? Yep. And can you click to biodiversity genomics galg inputs JSON? Yep. So this is our definition of the input items values. We used this file as the definition of the test data when running WDL files on local. And can move back to the top page again. And yeah, we have a make file, and we have a subdirectory name is first K, gene scope FK, Mercury FK. This is the directory of Docker container as we there are no Docker containers so far, so we created the Docker containers by ourselves as a temporary workflow. So you see some Docker file and some make file for convenience. I think, yeah, I explained everything. Basically, no, not everything. Sorry. And
the main things. Yeah.
yeah, but yeah, I want to.
And I would like to show that, like, for example, when you create a Docker container for each tool, you will see like those Docker containers in this Docker store as well. Like, for each tool, we contain this Docker image. So you also see this tasks ID in a Docker store.
Can I speak other things for the repository again? Can you click input directory? So here the test data, because the reason I wanted to share this information is you can create your own Terra account and you can run this workflow on your Terra account. And you need test data, obviously, right? So I wanted to tell here, this is some test data, valid test data to run the workflow. And yeah, test hi-fi fastq and test hi-c R1, R2 fastq. Those three files are important to run. Okay, so move on to the top again. And also, can you click Terra data directory? And click sample tsv. Yep. So when you want to run this workflow on your Terra account, you need test data. Test data means we saw test files in input directory, but the Terra provides the feature to run some table format test data. And you can see some first column is some ID. It's the ID of the data. In this case, we use the sample ID. And then second column is input file. It's a hi-fi file, right? And you can see gs colon // and the ending, this path ends with test.hi-fi.fastq. So this is the file path on the cloud, the Google storage. And when you create your Terra account, you need to upload the test files first to the Google Cloud. Then you need to replace this path with your own test data cloud path. And
maybe this is a good time actually to show this Terra account. I'm gonna open that now.
Okay. But actually, don't worry. We create the specific wiki page to for you to run this workflow on your Terra account. So later we will share that page.
Okay. So I hope you see now this Terra website.
Yep. Okay. So yeah, let me maybe introduce from workflow, I guess, or test data.
Yeah, I just clicked test data as, so the first step is you have to give an ID for the data for the sample. So here we chose S06. You can choose any you want. And
Just
now,
a moment. First, we need to explain about other data files. Can you click other data files link? Sorry. Data and other data files.
Yeah.
Click other data and files link, files folder. Yeah. This. And we see 3 test files. When you create your Terra account, you need to upload 3 test files to this to here. Okay. Then we click data tables, I guess, the sample. And we already created this table styled data, but if you create the data initially, first you need to click import data left side button. And then you need to upload your TSV file. Okay.
Okay. And then we go to workflows.
Yeah, okay.
And here,
Okay. Just a moment. Okay. And yeah, let me explain some items on the page. The first version, this version means our GitHub repositories main is the main branch, and other things are tag information on the GitHub repository. And then we need to select the how to use the test data. Yeah, and yeah, we already said in this case we already used from sample S01 to S04. At this time we will use S05, right? And
S06, I think.
yeah. Sorry, S06 we choose it. Okay. And of course we can choose multiple rows, but in this case just we choose one. And input, you can see input tab. This is the definition of the input items on the main WDL file. We saw it, right? And the attributes is the mapping from the WDL output items with table columns. And in this case, we just use same name. We did set same name. So this something name means a TSV files column name. And next we see outputs tab. This is a definition of the output items we saw on the main WDL file. Its variable is the item name in the WDL file, and attribute is the mapping. It's the same with the inputs. So it's we define new column names here.
So in this, essentially, you can get the output from any tool that you have used in the pipeline.
Yeah.
So you have outputs from FastK, you have outputs from HiFiasm or PS tools. So you can see outputs from any tools you have used in the workflow, in the WDL workflow.
Okay.
And this scripts is essentially the same WDL file that we saw also in the GitHub. And then I think now we can run the analysis. So we now submit the job given on this test data. That is, we can run, right, test run ECCB to W08, and then you can submit a job. Yeah.
Yep, we can see job history. And in our previous test, it took around 4 minutes.
So any questions or comments?
I don't see any questions, comments. Yes, I have a question for you, audiences. So, are you interested in using a workflow? Are you using workflow, or are you using? Are you interested in using a Terra platform? Live Q&A. Paul.
Okay.
So,
I think it's done.
okay. Can you share your screen?
Yes, I am, I think. Yeah, I am sharing my screen. So now so do you see my screen? It's actually the
Yeah,
page.
I can see it. And can you back to the some job history page again?
Yeah.
Yep. Okay. So now you can see the top row is the green and the status is done. So, but if you see some errors, just in case, can you click some left column, some left green color? Yes, yes. And you can see the detail of the job history, and I think this page is useful. And you can see some when you started this job and when you finish the job, and also you can see some three icons on the bottom area of links. Links and three. You can see three icons. Especially, yes, this job manager is useful because if you see one error, yeah, you need to log in. But yeah, and if this is a detail of the job history, and if you failed the error on the summary page, previous page, you can see the reason, cause of the error on the detail page. And as Terra is using Google Cloud internally for their service, this page is to manage the used files for the workflow job. And this page does not save every files created on a process. Rather, this page is only saves specific files that specified in WDL file.
For example, like the assembly that is generated at the config level is this one .far that you got, and then you apply PS tools to it, and then you can go back to the output of PS tools.
And
Wait,
we also included targz file of the major command as an output. So we can see necessary data files from the test data page.
so okay, for example, for the scaffolding step also, you whatever outputs you received from PS tools, so you can actually see them. These are in this temp folder. So this will produce two haplotypes, and as you see, there is this hap1.fa and hap2.fa. You can download them and easily and saw that. So, do we have anything more?
I think we can share the test data again, some created new test data tab and table and sample, and we can see S6. And before running the workflow job, there are only input columns. Output columns are empty. But now we can see some field value. This is
Like for example, the output from FastK, that is, and then you can easily down that this is the output from FastK, how many Kmer, it essentially gives the Kmer spectrum. So you can get Kmer tables and you can see the stats here.
right.
And similarly for other tools like for scaffolding, you got these two output files, and then for Hi-Fiasm you got the output. For GeneScope you got this output. So every step, whatever in outputs you receive, there's so you can visualize them in this table. So it's overall a nice workflow descriptive language, and it's very easy to run on Dockstore, and it's quite open source. So like on the first time you can make a free account on this, and then you can run multiple jobs for some limited period of time. Okay, so maybe that's all for the practical session. Thanks, Jun, for walking us through this nice Vidal platform and Terra. So maybe if you have questions, just write in the chat box. We'll be happy to help and answer. Um, and then now we are almost reaching towards the end of the workshop. So some last comments. First of all, we are planning to uh, prepare a manuscript related to this workflow, and also the topic is the same as the workshop. So if you are interested to contribute towards the towards the biodiversity, either in using this pipeline for different types of genomes. For example, if you have a new data that you want to run this pipeline on, or you want to contribute towards the software development part, that would also be. So any sort of contributions, or any guidance that you need, how to use this pipeline, you are encouraged to write your name and other relevant information in this spreadsheet that I just shared with you on the chat box. So if you are interested to contribute in any form, that would be encouraged. I hope you see this link, and I hope you can open this link.
Yeah, I can open it.
Okay, so take your time. We still have 15-20 minutes. So if you are interested in contributions or want to learn or use, then put up your name. We'll follow up on that.
If we have time, maybe it's good to explain about the dog store. Can you some open the link I pasted to the chat box?
Okay. Or maybe, or would it be good to explain doc store at the next level,
Yeah.
or the GitHub, or do you want to do it now?
Yeah,
I was
maybe.
thinking to close the technical session now and like have
Sure,
more
okay.
feedback, and
No problem.
let's do it like on
Yeah.
the subsequent meetings.
Yep, agree.
And also any feedback you have on the workshop, any that would help for in the next years, feel free to share. So the question is it possible to use the Vidal workflow other than Terra? Yes, I think you can run Vidal directly on Cromwell, right?
Yes, so actually, yeah, you even don't need to use the cloud. You can run a WDL file on your local, because just the engine is Java library, so you can run on local.
Yeah. And the good thing about Terra is initially it provides a free account, and it gives you some credit points to run some data.
Yeah, and yeah, customer support is very helpful. We
Yeah,
asked
so
many
like
questions.
yeah. So in that sense, like for a few genomes, if you want to assemble initially, you can use Terra and it's free of cost.
Yeah.
Initially. And it's quite fast because underlying storage and everything is with GCP, Google Cloud.
Yeah, you will have a 3 months free account with some enough amount of deposit.
Yeah. Any more questions? Feel free to ask any question. Don't worry about. Don't be hesitant. I think if we don't have further questions, we then thank everyone for joining today. I hope you have learned a bit, learned about these new technologies and new workflow language, and I hope this will be helpful in your research. And if you have any questions, if you are further interested in learning or contributing towards this topic, so just feel free to reach out either via email or just or create a pull issue or any issue on this GitHub page. We'll be happy to help you. Yeah,
And
so thanks
Silva,
everyone. Yeah.
Silva, thanks for some organizing this event and to have an opportunity as a speaker. So thank you very much.
Yeah, thanks, John. And thanks everyone for joining us. And many thanks to ECCB organizers, Paula, Julia, and everyone and Laura for helping with this event. It's been truly an honor to do this. Thanks, everyone. Bye.